It's 8:30, so I am handing it over to our session chair, Amy Maas, for a session on allometry.
Hello, everyone. Welcome back to Thursday morning. Bright and bouncy. All right, let's see. Button and advance. Okay. So I'm starting off our session. So we have 4 speakers after me, and I'm starting off with the basic premise that you had to look up allometry on Google when you read about this. So bear with me, we're starting from the beginning. And the reason we're talking about allometry is it's one of the ways that we go from process studies up to these big predictive things. And we use it because it works, except for when it doesn't. Okay, and so this is allometry, and the idea behind allometry is that there's traits, various things, that scale in a predictable way to size and often not linearly. Yes, these are beetles. No, they do not live in the ocean, and that's why this is cool. You don't need salt water for it to work, right? It's at big scales. We can see how body size scales with different components of the body in different ways, and that sometimes there are these traits that don't behave in a linear fashion. So these are the horns. There's sexual dimorphism. The females do one thing, the males do another thing because they're doing their peacock business and trying to fight and attract. And so there's a lot of relationships that we can use and model and think about and understand how this relates to energetics, okay? And the focus of this session isn't just sort of body shape, but also how size relates to rates. Because as we've heard repeatedly in earlier sessions, that's really what we're starting to realize we need to understand. How do rates respond to different forcings? And so the idea behind allometric scaling is that metabolic rate relates to mass based on a constant in some scaling exponent. We could spend an entire session with people, terrestrial, marine, limnology, who are thinking about what the exact relationship is between these things and how that particular equation should look. But we're not going to do that here. But the easiest thing to say is that as you are smaller, notice these are on a log-log scale, we have the size of an organism based on its metabolic rate. And if you're really small, you're a mouse, your heart rate and your metabolism is really, really, really fast. And as you get larger, things move at a slightly slower pace, not just in a linear fashion. It's a power law. And the reason that we think, argue, discuss, ponder about this is we think it has a bit to do with physics of nutrient delivery diffusion. Like George said, this is one of those gravity things that organisms have figured out a bunch of different ways to deal with, but this is a physical law. We have surface area to volume, that's true. Diffusion and entropy are a reality that systems all have to experience. And so allometry sort of captures some of those physical properties, and different evolutionary lineages have solved this problem in different ways, but they're still held to this rule. And so why do we use allometry? We use it to scale up. As you all know, ocean science is big science. It takes a lot of money. It takes a lot of effort. We frequently don't want to be away from small children, personally, for like more than 40 days. They get weird. They forget you. And so we have these limited time and money to make measurements, and we want to predict extremely complicated patterns because we're facing climate change, and we want to know what our future is going to look like. And so to do that, we take these single measurements and we want to extrapolate up to the community. Allometry helps us do that. Allometric scaling helps us make those changes. And full disclosure, I'm part of the exports program. I'm going to use export slides for the rest of it, and it is totally lobbying. And I apologize. Everyone uses allometry, but this is the data sets that I have available. So what I'm showing right here is what we're using allometry for. So this is the dry mass of a particular zooplankton. Each of the different colors is a different critter that we put into a syringe, and we measured its respiration rate. And so, there we go. And so they follow allometric scaling rules when you take them all together. We take those individual data points, and then we can go back to our communities where we take individual counts of bugs, we look at their size, and we say, yes, this is our community respiration. This is how much the zooplankton are contributing to the apparent oxygen utilization. This is how much active flux the community does, and we base it off of these allometric relationships because it's kind of impossible to measure the individual respiration rate of every organism. More than kind of, it is impossible, absolutely impossible. Other things we use it for is we use it for things like grazing rates, which I sort of think of as second level relationships. We have the predator size, so this is spherical dimension, diameter. These are our hypothetical spherical cows, except for there are hypothetical spherical flagellates, ciliates, rotifers. I think that actually works better than cows. They're slightly closer to spheres, sometimes. So we have their size, and on this side, this is the grazing rate. And so there are these relationships. You can see that there's quite a lot of spread in the data, but at least the broad paintbrush strokes, grazing rate seems to relate to predator size. And we use it for things like just basic, trying to figure out how much carbon is in the system, right? So this is very much surface area to volume. These are all data sets from exports. This is the biovolume of a bacterial cell and how much cellular carbon is in there. Fecal pellet volume, these are salps. We're all about the salps. And this is how much carbon is in a salp fecal pellet. This is the individual biomass of a particular zooplankton. This is how much is in there. So you see that these linear or power scaled relationships hold true across a lot of data sets, which makes it easy to scale up. We also do them for things like sinking rates. So this is the biovolume of a salp fecal pellet. This is how fast it sinks. And then, once we have these relationships, we use it to model. Okay? So this is Adrienne Bird's graduate student, Chandler Countryman. So she's working on a model where we take the size of a calanoid copepod. It's hard to see because I wanted to put everything up here. Size is on this axis, and you have ingestion rate based on size. Size is on this axis, and egestion rates on this. And you can put in the size of an organism, pull out, based on allometric scaling, what you think is going to happen. And then you can plug this into a model of vertical migration about where these critters are pooping in the water column and how much is coming out. Similarly, Kevin Archbold here, and a bunch of other people, were working on a model looking at small phytoplankton versus large phytoplankton, small zooplankton versus large zooplankton, where they're going to migrate and what that's going to mean for active flux. So we're plugging these relationships about the size of the organisms, the prey field and the predator field, and we're trying to say something about these bigger things, such as the movement of carbon from the surface to depth. And the reason we're having a session, and this is not just like, yay, allometry is awesome, is because we do have big changes in actual measured data points that may really matter, okay? And so some of these data points can be ecosystem level. So what I'm showing right here is mass on this axis and metabolic rate. So these are those metabolic relationships that we know hold really strongly across organisms. And this is all oh no. Backup button. These are all cephalopods, all squid, octopus, cute little squiggly bits. And yes, the line is there, and yes, the line follows scaling, but we have orders of magnitude difference in the metabolic rates of these organisms across just one taxonomic group. Some of them breathe a lot, some of them breathe less. And if you drill down into these things, we see that there's taxonomic and habitat evolutionary history differences in metabolic rates. So squid that are fast surface squid that live in oxygenated waters have these high metabolic rates. And these are fish with a very different slope because they have different physical constraints on their body type. This is a deep sea fish, and this is a deep sea cephalopod, the vampire squid, who just really sits mostly all the time and doesn't move around a lot. And so their metabolism ranges a great deal. So if you're just taking their body size, you're not going to be able to predict exactly what the metabolic rate is, even if you get the whole cloud and your scaling holds. Another reason that we think this needs more thought is that even within a single group or a single individual or a single size class, we see deviations due to developmental stage, right? Like small organisms that are about to go through metamorphosis respond very differently to situations than adult, robust organisms. They may be biomineralizing differently. They may have different energetic resources. And substrate stress is another major thing when we think about allometry, when things are not going to follow. So this is, again, work from exports. This is the small size class phytoplankton, larger size class phytoplankton. And this is how much chlorophyll is under these different nutrient conditions. So this is the same community held for 24 hours, and they're showing very different allometric relationships, chlorophyll relationships, and the communities are responding differently. The size classes don't respond to the nutrients in the same way. One of my favorite pet thinkings about right now has to do with how maybe sometimes size is just a covariate, right? That when we talk about the size of an organism, maybe apparent size might matter substantially. If you've got really long structures, your sinking size is going to be, your sinking rates are going to be different, but what if they're made out of something heavy versus made out of something light? That's going to matter. The other thing for this is gelatinous organisms in the ocean, right? There's a paper called Faking Giants about how gelatinous organisms, their surface area to volume versus their dry mass is very, very different. And so their ability to interact with prey fields is different, and their ability to deal with carbon is very different than copepods, which most of our models are based on copepods because they survive everything that we do to them, right? And so all of our relationships fit really well for copepods because that's where we made the measurements in the first place. And so a lot of these gelatinous organisms, which we're starting to understand are really, really critical, have different patterns that we need to investigate a bit more. So a larvation actually, for those of you who are not zooplankton geeks like me, teeny tiny little critter closely related to us, with a tail, and they build these huge houses and they move water through it, and they're able to encounter prey of a larger variety of size classes, and they can consume a lot of primary productivity. So the apparent size of a larvation is actually much bigger than the critter itself. Wrong button. Another reason that this is still a conversation is because our science is so expensive and time consuming and blah, 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 blah. We have this data point and this set, and we measure it and we make it fit, and we're like, yes, we've got an R squared, and then we try to go to our observations of the whole global system, and then it doesn't work. And so we're missing something in a lot of our predictions. And what I wanted to show is some work relating chlorophyll A, so rating the stock of phytoplankton with grazing rate. And there's none of these relationships, even though we know that there are these underlying allometric things that should hold weight. Same thing can be said, this is Deb and Mike Landry's work, mean migrant biomass and respiratory flux. So biomass to these other processes aren't working. And one of my bits, I'm like, so therefore it's not working. Or maybe just measuring stocks, which we've been doing for a long time and we do well, that's not enough, right? Because we do need to put traits in, whether those traits or size or other things. Okay, and why I think that's important now is I'm feeling like physiologists, process studies people recognize allometry and traits are important. And the conversation that I hear from the modeling community is there's an understanding that traits are important, but we can only handle so many, so let's figure out the right ones. But a lot of our observations are still based on stocks, again. And this is the shout out for, this is why we need PACE. This is why we need more instrumentation in the ocean that's looking at more complex variables than just our stocks. And we have a lot of new systems that are going online that are becoming part of our continuous observations, that are becoming part of our time series, that can measure these sizes. And they can measure them on a much more continuous way, much less expensive way. And these are just a few systems that are out there that we're implementing to look at organisms' size classes across the scale in the ocean that we can then funnel that information into our models and our understanding. And again, plugging again for things that I'm doing, but we have these big observational programs ongoing right now whose entire purpose is to go from process studies and move up to being able to model. And all those data sets are supposed to be mandated, they are mandated, to be available to everyone to start working with, so you can start looking at your process study of choice and how that relates to these larger observational data sets, satellites, et cetera. Because I think it's really a good time to see how much allometry actually captures, and then to find other traits that are important for covarying and then implement them. Now, I've used the word trait a lot when I'm talking about things, and I think that that's an important thing for us to mention and talk about for those who aren't familiar with it yet. So a trait is just anything that we can use to talk about life history, ecological or biogeochemical niche. So size itself is a trait. It's sometimes been called the master trait, and I'm hoping some people will disagree. They might be alone in this, but for your pet particular thing, size might not be the dominant thing. Depth range could be used as that trophic position, whether you're a biomineralizer or not. Those are all different kinds of traits that we can assign to organisms and see how it co-varies with ecological niche or role in the community. And my goal for this session is then to basically expand the usefulness of allometry to other applications. And we heard this actually talked about a bit in some of the earlier sessions when we were talking about expanding hypoxia, when we were talking about nitrogen loads. There was a lot of conversations about systems and scale and retention time or circulation and how these things relate. And we have a speaker who's going to talk about ways that we can think about allometry applying to things other than just single organisms. I think another goal is to try to find some of the second level traits that capture the variability that contribute to our ecosystem function. And for me as a physiologist, size really is a nuisance variable. I have to measure it because I know it's going to co-vary, but it often masks the thing that I'm interested in, which is response to ocean acidification or response to low oxygen or response to temperature. And then there's sometimes when it's not really driving the observed pattern that I'm looking at. And I think it's important to recognize when size isn't actually the driver, it's just along for the ride. And so the way that we do this is we can, and this is where I think now is the time to talk about allometry again, because it's not like this is a new idea, but we're in a place where we can really start looking at the crosstalk between models, process studies, in situ observations, because all of these things have moved to a new phase. And I want to look at those mechanisms. So, your job, you don't have a choice but to choose it because you're stuck in this room, is to think about your thoughts about allometry. Think about ways where we can use it more because it is effective, and where we should use it less. And then alternative traits or applications that could improve our understanding of biogeochemistry. And I left lots of time for questions if I've made things unclear rather than clear.
Oh, Alison MacDonald, Woodsall. I'm a physical oceanographer. I know nothing about this. But why is it that the way you presented it, that it's size versus one thing? Why isn't it size versus 15 things?
It is size versus 15 things. Absolutely. Okay. I think because as a physiologist, my understanding is that I'm not allowed to get as excited about the details, every single physiological thing that contributes to the variation, because the model can't handle it. And so I think that each function that we're trying to apply can probably have like 2 or 3 components that we think about. And what I would like to recognize is that they're not necessarily going to be the same thing for all the different rates or functions that we're looking at. So size tends to be the top thing. What that second or tertiary trait or covariate is going to be, I think may very much depend upon what question you're asking, right? And so I wanted to leave that open to other things, because it's not going to be just one thing.
So you talked about size, but do you think there are other traits that, what are your ideas for other traits that we should be thinking about?
For me, I think some of the other things is this feeding strategies, which I think encompasses a lot of size scales. I think the strategies of the smaller levels, mixotrophy, diazotrophy, and then when you get to larger size classes, whether you're a sit and wait, hang out and do nothing kind of organism versus a very active organism, whether you're a filter feeder that builds, or something that builds a house versus a copepod which passively filters. I think that that's a really important thing for the kinds of questions that I'm thinking about. I also think that if we go to this idea of like the evolutionary history of organisms, that specific groups don't have the capacity to in our models, everything is possible because it's the whole community, but certain organisms have only certain capacities. And so I think ability to deal with hypoxia, so I'm a zooplankton person, so I think about the zooplankton. Ability to deal with hypoxia is going to be a big thing. Those are the things that I'm thinking about, but I'm seeing, and when I hear things over and over again, I hear feeding trophic level, and also how well you recycle. Whether you're something that just is like a salp who like vacuums up the world and just poops and is like capitalist in the way that it works, versus something that's like, oh my gosh, there's not a lot of phytoplankton here. If I turn it into big fecal pellets, I'm not going to have any poop. There's not going to be any regeneration, which is sort of more of a sustainable culture organism. Then those kinds of strategies, and that's not just organismal level, that's also how the community responds to the nutrients availability within a particular region.
Chris Sabine, Hawaii. So is there an implicit assumption in all this that these allometric relationships are constant? Or do they change over time? You started off showing a picture of bugs, and I immediately thought of the giant bugs in the Cretaceous that...
With more oxygen.
Yeah, so is it possible that these things are changing?
I think so. And I think when we were talking about trying to get the rates correct for photosynthesis and respiration, we're talking about like, are those going to be responding differently? Are the allometric relationships going to be responding differently in a changing world? And are they going to be responding differently to temperature, right? So I think they will. And we have to be careful not to just say, okay, quarter power rule, perfect. And that's why for me it's a nuisance variable. Because yes, of course it's going to covary. But how many data points do you have to take before you realize that the covariation is changing, right? And so then in some ways I think it's a question of having enough observations to be able to see that. So.
Hi, this is Jamie Pelter from Rhode Island. This is just a curiosity question about, I'm aware that there's like the Darwin model for phytoplankton at least that encodes traits, not size, and then runs and we see what happens. If you go back and then look at it as if you only had size information, can that give us insight about how?
I would love for Steph to comment on that.
Do we have can we? Say it again, so everyone can hear.
So I think I agree that size is not the only trait. I think you have to have other traits as well, and you can turn them on and off, and you can get very different results. So size is not the only important trait.
But she did say she can run it just using size.
Yeah. And not just using size,
Yeah.
right? Yeah. So for instance, I can run with functional types like diatoms, diazotrophs. I could run it with size. I can run it with just trophic strategy, and you can get very different results, very different distributions. So I think you need all of them in there. Size is actually, size maybe actually is one of the, but as soon as you take things away like diazotrophy or mixotrophy, you get big changes as well. So.
It's the yes, yes. We're really smart people. We can hold two thoughts in our head, right? This is the yes, yes. Like I hate size, but I also know I need it, right? Like. We're out of time.
Oh, that's fine. I went through and looked and it comes out okay.